Welcome to The Explainer. Today, we're going to crack open the Medical Detective's Toolkit. We're going to see exactly how pathologists solve these incredible biological mysteries, starting with just a single tissue sample and ending with a diagnosis that can literally change a life.
So, it all kicks off with this one simple question. A doctor removes a piece of tissue. Sometimes it's no bigger than a grain of rice, but that tiny sample, it's about to go on an amazing journey. It's a super sophisticated process that takes it from the patient's bedside to the lab and finally under the pathologist's microscope, all to unlock the secrets hidden inside.
All right, so here's our roadmap for this explainer. We're going to follow the evidence step by step. First, how that tissue gets prepared. Then, how we read the clues from the cells themselves. Next, we'll look at their molecular fingerprints. And finally, we'll see how it all comes together for the final diagnosis.
Okay, let's dive right in. Our first stop is all about understanding the fundamental journey that every single tissue sample has to take. And trust me, this preparation is meticulous, because without it, a diagnosis just wouldn't be possible. You're looking at a 6-step transformation, and every single step is absolutely critical. It all starts with the pathologist's own eyes, what we call the gross examination, where they look at the whole specimen to find the most suspicious parts. Then that tissue gets fixed in formalin, which basically freezes it in time so it can't decay. From there, it's processed into a solid wax block, sliced thinner than a human hair, and finally stained. That classic pink and blue H&E stain, that's the workhorse of pathology. It turns the cell nuclei blue and the cytoplasm pink. It's only after we turn living tissue into the stable, color-coded map that the real detective work can begin.
So, now we have a slide. This is where the real investigation starts. And the first level of analysis is called cytopathology, where we zoom in from the whole tissue and focus on the individual cells themselves.
Cytopathology is really the art of reading clues from individual cells or maybe small clusters of them. You've probably heard of things like pap smears or fluid analysis. That's cytopathology. It's often done with techniques that are minimally invasive, like a fine needle aspiration. So why is that a big deal? Well, it lets doctors get a crucial first look at a suspicious lump or a nodule with very, very little risk to the patient.
Okay, this is a really crucial point, the difference in the evidence. Think of it like this. Cytopathology is like looking at a police lineup of suspects. You can see if they look suspicious based on their individual features. But histopathology, where you're looking at the whole tissue biopsy, that's like examining the entire crime scene. It shows you the relationships between all the cells and, most importantly, if any malignant cells have started to invade the surrounding area, and that is a key piece of evidence for a definitive cancer diagnosis.
And this table, it brilliantly shows how cytopathology is used in the real world to guide patient care. The Bethesda system for thyroid nodules takes that cellular evidence from a needle sample and translates it directly into a risk of cancer and a clear recommendation. So, a Category 2 result means, hey, just routine follow-up. But a Category VI finding, that means definitive surgery. It's a perfect example of how pathology directly informs what happens next for the patient.
All right, let's move on to our next set of tools. You know, sometimes what a cell looks like just isn't enough to solve the case. We need to know what it is on a molecular level. This is where we go way beyond just shape and color to identify its unique molecular fingerprints. And these can tell us everything from where a cancer started to exactly how to treat it.
The first tool is called immunohistochemistry, or IHC for short. The best way to think about it is like a set of incredibly specific molecular highlighters. Scientists create these antibodies that will only stick to one very particular protein. So when you apply them to a tissue slide, they latch on, a chemical reaction makes them visible, and boom, you've effectively color-coded the cells, telling the pathologist exactly what they are.
And here's what that looks like in the real world. Say a pathologist finds a tumor in the lung. They can stain for a marker called TTF1. If that stain is positive, it strongly suggests the cancer started right there in the lung. But if it's negative, it might be a metastasis that traveled from somewhere else. Now, why is this so critical? Because the treatment for a primary lung cancer is totally different from, say, a colon cancer that has spread to the lung. Other markers like ER/PR in breast cancer don't just diagnose, they actually predict whether a patient will respond to certain hormone therapies. So it's diagnosis and treatment guidance all wrapped up in one test.
But what if the clues aren't in the proteins? What if they're hidden even deeper, inside the cell's genetic code itself? Well, for that, pathologists turn to a technique that lets them navigate the genome directly.
This amazing technique is called FISH, which stands for fluorescence in situ hybridization. Basically, it uses these tiny fluorescent probes that are designed to stick to very specific sequences of DNA. So when you look under a special microscope, these probes light up, letting pathologists literally see genetic changes that are otherwise completely invisible. We're talking about a gene that's been copied too many times, or maybe two genes that have been improperly fused together.
Now, what's so cool about this is the direct line you can draw from a specific genetic finding to a targeted, often life-saving treatment. Finding extra copies of the HER2 gene in breast cancer, that means the patient is a candidate for Herceptin. Finding an ALK gene rearrangement in lung cancer, that opens the door to using ALK inhibitor drugs. This right here is the heart of personalized medicine, all driven by molecular pathology, where the diagnosis itself contains the blueprint for the cure.
Okay, so after gathering all of the evidence, from how the cells look under the microscope to their unique molecular fingerprints, the final step is to put it all together into a comprehensive verdict. And this isn't just a label, it's a detailed report that's going to guide the patient's entire course of treatment.
Two of the most important concepts here are grading and staging, and it's really important to know the difference. Think of it this way. Grading describes the tumor's personality. Are the cells well-behaved and look kind of normal? That's low grade. Or are they chaotic and aggressive looking? That's high grade. Staging, on the other hand, describes the tumor's physical footprint. How big is it and where has it traveled in the body?
And the gold standard for staging is something called the TNM system. It provides a simple, standardized language for every cancer. T stands for the size of the primary tumor. N tells us if it is spread to nearby lymph nodes. And M describes whether it is metastasized to distant parts of the body.
So here's the bottom line. While a high-grade tumor is definitely dangerous, it's the stage that is generally the single most critical factor for a patient's prognosis and their treatment. I mean, think about it. A small, high-grade tumor that hasn't spread might be totally curable with surgery, but a low-grade tumor that's already spread everywhere presents a much, much bigger challenge. Knowing exactly where the cancer is located is everything.
So we've gone from a simple piece of tissue processed into a slide to reading cellular clues, to uncovering molecular fingerprints. The Pathologist Toolkit is, without a doubt, a powerful engine of modern medicine. But now, with AI that's becoming capable of analyzing these slides, it leaves us with one final big question. What does the future of the medical detective actually look like? Could it be a partnership between human expertise and artificial intelligence, one that's poised to unlock even deeper biological mysteries?